Well, good morning. How's everybody doing? This is really exciting opportunity to come and talk just a little bit about some pretty exciting research that we've been doing over the last couple years in the Intermountain regions. Today I'm going to talk just a little bit about some research we've been doing to understand some of the drivers that influence our restoration outcomes in terms of native plant seedings. Erica did a really nice job earlier on talking about the need to think about processes that influence what happens on the landscapes, not just talk about species lists or tools, but actually trying to manage processes that influence restoration outcomes. And what I want to do today here is talk about particularly the role of winter conditions. So just to set the context for this a little bit, so I'm now at the University of California, but I spent a good 7 years in Eastern Oregon and other parts of the Intermountain region working on seedling establishment issues. As I was driving down, literally I had exactly 10,000 pounds of stuff because they weighed it, which is really awesome to find out you own that much stuff. We drove through things that looked exactly like this last summer. You guys probably all remember these. So these are multiple fire complexes throughout the West that were burning simultaneously. Huge amount. I mean, we're talking about 1.4 million acres or more total that got burned. A lot of animals got burned. Huge hit on the agricultural community. Huge impact on our natural resources. We know after catastrophic events like this, a lot of even our fairly intact rangeland can look really degraded. We do know that these rangelands provide a host of important ecosystem services. So it's not just agricultural production, but a whole suite of services that society gets from rangelands. We know if we don't intervene, in many cases, our rangelands will rapidly convert to something like this, maybe dominated by cheatgrass and Medusa head. One of the, salient syntheses that's been developed in the last couple of years has to do with the NRCS conservation benefits assessment effort, where NRCS, basically through a charge for Congress, was asked to evaluate if the conservation practices that they spend money on are yielding the anticipated conservation benefits. Some of you might be familiar with this synthesis effort. It's a multi-chapter book that looks at a suite of rangeland management practices. One of the chapters had to do with seeding and rangeland weed management. Basically asked if we're spending these conservation dollars on it, what are the conservation benefits that we think we're getting out of it? A comparable, or at least a linked synthesis effort through the GAO that was done several years before that asked some of the same questions. And what they're basically asking is, we have a set of conservation practices that we do after fire, weed management treatments, and so forth. What are the outcomes of these practices? And probably not surprising to most of us in the room, when we're talking about native plant seedings, Medusa head management, cheatgrass, it turns out that on average, most of these restoration efforts fail when we're working with native plants. It's hard to get a ballpark number around these, but it wouldn't be surprising if we're in the order of 5 to 10% of our practices are actually successful when we're using native plants. The bottom line with these is that as we have changes in fire frequency, spread of weeds, impact on our rangelands, probably 5 to 10% of our restoration efforts using native plants are successful, and we're spending a whole bunch of money on this. So it's a pretty serious issue. We've pretty much been aware of this for many decades, and for many decades we've been putting a substantial amount of research and management to try to enhance these outcomes. But I think the reality that most of us could agree in this room we are very, very far from being where we want to be as a field. So one of the lines of work that we're trying to pursue to better understand what drives these outcomes and enhance our ability to manage these outcomes has to do with really understanding what goes on during seedling establishment following, let's say, a fire or a weed treatment. And the idea here is very simple. We want to know when do our native plants, or how many of our native plants die when we seed them? When do they die? And if we know when they die, we can start identifying ecological mechanisms and processes that are causing them to die. If we can identify those processes, then it's going to be probably pretty reasonable to develop some management solutions to mitigate those processes. So this line of work is very simple. It's very intuitive. We're just asking, if we go out and put native plants in the ground, when do they die? How many of them die? What's causing them to die? If we identify what's causing them to die, we can probably get some insight about what we can do different in terms of management. So just to give you a rundown real quick, when we go out and drill native plant, introduce plant seeds after a fire or weed management effort, this plant material is going to go through several major key life stages before it becomes an adult plant that's able to produce seeds and facilitate recruitment on its own. So we put seeds in the soil with a drill, broadcast, aerial seeds, something like that. Those seeds that we incorporate into the seedbed need to first germinate, so that's simply just producing a radical or shooting a root system. That germinated seed needs to emerge. So this is a picture of a coleoptile. So this is a grass seedling. This is less than a centimeter here. This is the first step where you can actually see the plant above the soil surface. After that coleoptile or cotyledon emerges, the plant needs to produce true leaves that become photosynthetically independent or photosynthetically functional, and it can make its own energy. That seedling then at some point needs the tiller, survive some juvenile phase before it's able to become an adult plant that produces seeds. So what we're doing in this first step is asking, so we put seeds in the soil, what are our mortality rates during each of these key steps? If we identify when most of our mortality is occurring, we'll be in a good position to identify the cause of that and then again our management. So I'm not going to take you through all the specifics of this, but this is summary data from Eastern Oregon across 4 sites, 3 years. We have introduced grass in the open circles and a native grass in the closed circles. This is survival probability on this axis, and on the x-axis here, we have our key stages. So germination, seedling emergence, seedling establishment, survival to juvenile, survival to adult. And what we see across most years in this part of the country with most of our natives and introduced plants, germination, so actually producing that radical, is fairly high. You know, we could be 70 to 80% of the seeds that we incorporate into the seed bed germinate at some point. But for our natives, only about 20% of those germinated seeds emerge. In some years, we've seen this number as low as 4%. In contrast, for our introduced grasses like crested wheatgrass, we have about 70% of those germinated seeds make it to that coleoptile stage. Through time, you can see that survival probabilities tend to increase, remain fairly high, and they're similar between these species types. So one of the major patterns we picked up, at least in Eastern Oregon, is that a large difference between what crested might be doing and, let's say, blue bunch or squirrel tail has to do with something going on during this life stage transition. So to put it in pictures, what we're seeing for our natives, about 90% of the mortality, the seeds you put in the soil, occurs between when you produce this little radical and when it gets here. So we're very interested, obviously, if 90% of our mortality of the things that we sow occurs in 1 stage transition, it's going to be pretty helpful to understand just what in the world's going on there. So that's kind of the key focus for the idea of what is going on over winter, and I'll get into that in just a second. But if we step back and think about, there's a suite of things that could be going on during that critical transition from a germinated seed to a seedling, or an emerged seedling. There's a lot of things in the soil that like to eat things that have germinated, so these are like fungal pathogens, Pythium and Fusarium. We know in our inner mountain regions, I mean, freeze-thaw cycles are substantial, wet-dry cycles are substantial. Any of that sort of physical moving around in the soil, I mean, that can be pretty tough on something trying to grow a root system as well. I know this is going to be quite hard to see for most of you, but what this shows is water content and temperature in the seed bed, so about an inch down through the course of a couple years in sort of a typical intermountain scenario. And again, this is what germinated seeds are looking like. What we're seeing in many cases is if we go and do, like, let's say a late fall seeding, so late October, early November, mid-November, sometime before Thanksgiving, in many cases we can get maybe some good fall rains, and during those fall rains we might have some periods where the soil and air temperature are actually fairly warm. We could have a 1 or 2 week period where we might get 50-degree days, right? That's not that uncommon during many of our falls. What we're finding for a lot of these bunch grasses, our native and our introduced, is these brief periods of warm, wet falls, even if it's around Thanksgiving, are enough to stimulate germination of these seeds. So what that means for practical purposes is that, let's see, here's like Thanksgiving time here for temperature. You guys can follow the dot. Here's the 0 line. We have a very, very long time that those seeds are exposed to temperatures as low as 10 degrees, minus 10 degrees Celsius. So a pretty tough environment and that the seeds are exposed to for a very long time. So the aim of this kind of work that I'm going to show you is to really try to understand what's going on during these winter periods. We know that germination is occurring in many instances during winter. So the obvious question then is, if many seeds are killed following germination and they're germinating during winter, what are the factors that are causing this death? And again, the idea is if we can identify the factors driving that death, we'd be in a better position to develop some management ideas. One other aspect to consider is physical crusting of soils that can occur during winter and early spring as well. Particularly our soils with high silt content. You guys have probably seen a lot of this plating and so forth. One of the ideas here that we wanted to look at is that if these soils are obviously wet and moist during winter, they don't crust, right? Soils only crust when they dry. So good wet winters with snowpack are probably going to have soils that crust later on and not as severely as perhaps dry winters. So one of the things that we wanted to examine, in addition to freeze-thaw cycles, pathogens, and things like that, is potentially the role of soil crusting and how that may or may not change under different types of winter conditions. So the basic experimental approach we took was to manipulate 3 major factors during winter. So one is we disturb plots. We just simply tilled them to alter the soil physical structure. That changes the crusting. So plots that get tilled, either with if it's a rangeland drill or just simply tilling the soil to prepare a seed bed, that disturbance on silty soils can change the soil physical structure and increase or change crusting. We manipulated snow during the winter to really look at the integrated effect of different snow levels on seedling recruitment. And we also coated seeds with fungicide to see if we can alter the pathogen levels on those seeds. And what we basically looked at is recruitment of blue bunch, wheatgrass, and cheatgrass in these 3 treatments here. This is what the plot looked like. This is up in eastern Oregon. And we actually had a very fortunate year to do this. One, we had snow, which turned out to be useful if you want to manipulate snow. And it turned out that, ignorance is really bliss in this, because all the year, like 2 years before and 2 years after, there's almost no snow cover. So ignorance is helpful in many situations. The other thing that was really helpful with this is we had a very, very wet spring, and I'll bring those numbers up again. This is 2010, 2011, so we had about 5 inches of precip in spring. So what this allowed us to do, and this is important when we're looking at just the nuts and bolts outcomes here, is it really allowed us to put numbers behind how do changed wintertime conditions impact your seedling recruitment, independent of what spring had. Because again, during spring, 5 inches of precip, soil's pretty much wetted up to field capacity, treatments were similar, we weren't manipulating anything in spring, so the differences that we see be attributed to what goes on in winter. So this is just some sensor data. I know this is really hard to see. Couldn't think of a better way to do it for a presentation, but we have soil water content on the top, soil temperature in the back. We have 3 levels of snow manipulation. We just let ambient snow did what it did. Some plots we removed snow down to 5 centimeters, so we left a little bit but took away much of that insulated layer. And then some plots we added snow. And this red line here is when we stopped doing snow manipulation treatments. So, things like soil temperature, soil moisture after this red line tend to stabilize to some degree. Most of our treatment effects, because this is when we were doing the manipulations, occurred here. So as one example here, this is the snow addition treatment, so it increased soil water content substantially during this period. This dark line down here that you see, this is snow removal. So during that same period when the soils were very dry in the snow removal treatment, they were also very cold. So I'm not sure what happened here. So the major point with the snow treatment, though, is you're not manipulating one factor. It's not just temperature. It's not just moisture. It's not just soil crusting. It's an aggregation of multiple things. So we're not at the point where we're going to assign one causal mechanism, but we're in the process of sort of narrowing down what's going on. So, Tom, is there something else that I should press? Okay. What we're looking at here is the fungal load on germinated seeds during emergence for cheatgrass and blue bunch. Just think of this as an index of the entire fungal community on those seeds. It's not just the pathogens, it's the entire fungal community. We're trying to work out the exact type of fungi right now, but think of this as an integrated index of the amount of fungi that are on the seeds. So for cheatgrass, when we removed snow, we did get an increase in fungal biomass on cheatgrass seeds. Really not a lot for blue bunch. Not a major change, at least that was significant. And the summary point here is even with the fungicide-coated seeds, we put fungicide on half of our plots onto the seeds. We didn't see any change in emergence for either cheatgrass or blue bunch. So at this point, there's not a lot of evidence that the fungal community there is causing a lot of that mortality for either species. Real quick with the disturbance, so these are plots that plated up really good when we tilled them. These are plots that are left undisturbed. Till control, this is penetration resistance. It tells you basically how much of the soil is crusting in these treatments. As the soils are fairly wet, differences are small. As the soil dry, differences become large. This is a well-established physical phenomenon, but basically it's just showing that between these two treatments, we did have significant differences in penetration resistance, and this occurs almost exclusively as those soils dry substantially. So this is the main summary version of the results. So we're asking the question, what goes on as these wintertime conditions change for our native plants and cheatgrass? On this axis, we have cumulative survival probability. We have our 3 snow treatments. So these are plots we took away snow. Snow is left as a control, just whatever came. And and the plots we added snow. And this is again for cheatgrass, blue bunch. Disturbed soils are black dots, undisturbed soils are white dots. So the general pattern we see for both our invasive cheatgrass or native blue bunch is as we add snow in winter, we have pretty large increases in total survival probability. In some cases, I mean, we can look at a doubling almost of survival probability from 10% to 20%. Cheatgrass was a little more muted, but again, if you just look at the differences in scale here, we could probably argue that whatever's going on in winter has some ecological implications. You know, if we're going to get a doubling of survival just based on what our snowfall is during that condition. Disturbance in most cases decreased establishment, but we can see that effect was often much more muted than just the effect of those ambient snow conditions. One other thing I want to point out here is we seeded the same number of individuals of cheatgrass and blue bunch in these treatments. So what this is also telling us is the per capita recruitment, or the number of seedlings that make it per number of seeds sown, is higher categorically for our native plant than cheatgrass. So regardless of these snow conditions, on an individual seed basis, blue bunch has higher recruitment rates than cheatgrass, which I thought was pretty darn important. So it tells us, we know in many cases cheatgrass might have 10,000 seeds per meter squared, and we're seeding 500 seeds per meter squared of blue bunch. But on a per seed basis, our natives can do quite well under a range of conditions. So nuts and bolts here. Wintertime conditions are looking to be critically important for both cheatgrass and blue bunch recruitment. This is despite, in this study, you know, we had 5 inches of spring precipitation. We had moist soils through May, so springtime conditions in this year did not limit recruitment. It was very directly retied to what we manipulated during winter. And as we manipulate these winter conditions, we can, in some cases, see a doubling of mortality, or conversely, a doubling of survival. We don't know yet if it's freeze-thaw cycles, wet-dry cycles, but the aggregated effect of snow and those environmental conditions during winter were hugely important in determining those outcomes. So the very reasonable question at this point is, if we have some sense that something in winter is causing a lot of our mortality, can we use that information to change what we do on the ground? This is a project that we're currently implementing now, just going to finish up with this, that is trying to test actually the effect of various management alternatives to manage or mitigate the effect of those wintertime conditions. So on this side here, this is the idea of coating seeds to try to mitigate some of those conditions. And there's a number of things that we can do here, but the idea here is that once we've identified the process or sets of processes that are causing the mortality, we can think very carefully about what we want to do to mitigate this. So Matt will be talking much more about this later. He's here today. He's he's way smarter than I am and is able to figure out exactly what to do with this stuff. But the key thing is here, we kind of know where we need to go, so we bring folks like Matt in that can actually figure out what to do with some of the seed coating efforts. Seed coating's been around since we could coat stuff with seeds, right? A lot of it's sort of coat something and throw it out and see what happens. But what Matt's doing differently is he's first identifying what we need to manage and then developing the specific coat to do it. So instead of just throwing, like, let's say, nutrients on it, he's trying to do stuff like agglomerate seeds so that collectively, instead of seeding things individually, collectively they germinate and can actually break that physical crust during those dry winters. You know, so very creative idea. He'll talk more about a series of this work later. But the important thing here again is we're at the point where we know some of the things we need to manage. Now we need to develop the tools to actually do that. And this is kind of how we're working through it. We could do things with some complex modeling to try to predict winters and sites that may be less conducive or more conducive to establishment. This is probably a little more technical and long-term, but again, a reasonable approach. We can do things with our plant materials. If we know that certain periods of the year are causing most of our mortality, we can identify plant traits that influence mortality and manipulate those traits if we need to overcome some of those processes. So just to kind of wrap up here, an idea I just want to extend an invitation to, we've established a series of sites throughout the Intermountain, and I know Lori's here today, and she knows where all these sites are, and is saying, oh my goodness, there's a lot of whatever those things are, pins on there. So we have about 15 sites throughout the Intermountain region where we're seeding the same seed lots, making the same measurements across a broad stretch of environmental conditions, and trying to identify how these demographic patterns may change depending on our environmental conditions. So the data I showed you just previously was really centralized towards this part in Eastern Oregon. It's a very fair question to ask, do we see these trends throughout the larger part of the Great Basin? And if we do see them or we don't see them, either way, the next question is, can we predict in a particular region what may happen in terms of seedling recruitment? And if we can do that, then we can potentially apply the appropriate management strategies at a particular site or at least a region. So that's where we're trying to head with this. There's a number of collaborative opportunities. If you guys are interested more in sort of what we can do with this structure, I'd be really interested in talking with you guys later about any ideas you had. So just to wrap it up here, this idea of seedling recruitment is a huge problem. It's at least in the order of $300 million a year, and probably at least around 20 million acres that we're dealing with, where we need to try to significantly improve our ability to establish seeds. Again, just like Erica outlined, what we're trying to do is link these outcomes to mechanisms and processes, not just a species list or a recommendation what we should do on a particular site, but actually understand the mechanism processes that influence those outcomes. And really what we want to do with this multi-state, multi-site approach is to be able to develop some predictions about what's going to happen in a general region. So if we do develop effective management tools, we know when we can deploy them most usefully. So that's it. If you guys have any questions on that?
Questions for Jeremy. Whoa. Sorry.
Yeah. It's Anatone. Yeah.
Anyone else? Okay, Andrew Polmatisky and Jeremy, your question.
Thanks, this is a great research. I was just wondering about last time Washington. I'll find that seeds sit out there for a couple of years, and then a wet spring, the blue button does really well that wet spring. And I'd be a little worried with your results that, in most of the other years where it was a dry spring, you would have had 4% blue button germination and 10-15% cheap grass germination. Does it replicate this in a drier year, or any thoughts about it?
With the wintertime condition manipulations, or just the general patterns of death that we've been seeing?
The general patterns of death, when we look at the germination.
Yeah, we've actually, right, Lori? We've repeated the heck out of this within the Eastern Oregon sort of region. So 5 sites, we're on our 5th year now of making these measurements. It's a small subset of species, so it's one type of blue bunch, one type of squirrel tail, and crested. We don't know anything beyond that. But what we've seen routinely in this area is fairly high germination, even in drought years. Not a lot of seed bank carryover, because we have 80% of the seeds we sow germinate that first spring. Maybe 5 to 10% of those die. They get eaten by something, some pathogen. So we're only talking a handful of seeds that would potentially carry over to the next year that we're seeing in this region. And then pretty categorically past that, a lot of death during that emergence step. So again, a limited region. And I think the question you pose is very, very fair. And that's what we're trying to get with this broader attempt, is to see if we see general patterns or if they shift significantly depending on, maybe areas in Washington with higher thermal time or something like that. Maybe the processes and death patterns differ substantially. So very fair question, and we, sort of limited targeted data set, but within that region, heavily replicated.
One more question. Okay, thank you both.
I was wondering whether you look at, consider the death that the seeds were planted and how that would influence the mortality of the emerging.
Yeah, excellent question.
Repeat the question.
Question is if we've looked at the role of depth and mortality in the field under these conditions, and we have not. We've basically sown everything at kind of the recommended targeted depth. So seed soil contact was good. We didn't really look at broadcast seeding or deep burial. Very good question, though. I think there's a lot of good work to be done in that area. Whole nother line of work, actually. Yeah. All right, well, thank you guys very much, and we'll probably want to move on. I'm